Hello, welcome to the Illinois Soybean Association Field Advisor webinars. Today is on red crown rot disease, which is an update in Illinois, and we're lucky enough to have Dr. Boris Camiletti, who is newer to the University of Illinois, and he's their extension plant pathologist, as well as Dr. Stephen Clough, USDA-ARS research geneticist and plant pathologist. I am multitasking today. I am live from the U of I extension in Macon County, so doing double duty here. My name's Stephanie Porter, and I'm the outreach agronomist for the Illinois Soybean Association. And as always, before we start, we have a few housekeeping items. So this webinar will be recorded and available after the live session. It will be emailed out along with the survey and on Field Advisor's YouTube channel. 1 CEU credit in pest management is available if you're watching this webinar live. A QR code will be available at the end of this presentation. However, if you're watching the recording of this webinar, we recommend self-reporting for your CEU credit. And if you have any issues, please email myself or others, Field Advisor, if you need assistance with the CEU credits. And then lastly, we're going to save the questions to the end of the webinar. And if you have them, I guess throughout the webinar, you're welcome to put them into the chat session in the meantime. But again, we'll get to the questions at the end of the webinar. And so now I'm going to get to the presenters and introduce them. So first off, we have Dr. Boris Camiletti, which it's his birthday today. Happy birthday, Boris. What a great present. Talk about red crown rot. He is assistant professor of crop sciences at the University of Illinois, Urbana-Champaign, and an Illinois Extension plant pathologist. He leads the SMART Plant Pathology Lab, focusing on plant disease biology, epidemiology, and sustainable management. His team integrates nanotechnology, remote sensing, molecular tools, and drone spray technology to improve disease control in soybeans, corn, and wheat. Dr. Camiletti, who earned his doctorate from the National University of Córdoba, Argentina, was lead inventor on a patent for nano-formulated fungicides and enhancing their effectiveness. So we'll start there. I'm going to go ahead and introduce Dr. Stephen Clough, if you want to start bringing up your presentation for us. Our second presentation after Dr. Camiletti is going to be Dr. Stephen Clough. He is a Bradley, Illinois native and is a plant pathologist and research geneticist for the USDA-ARS at the University of Illinois, Urbana-Champaign. His research focuses on soybean disease resistance, pathogen behavior, and host molecular responses. He earned a biochemistry degree from UC Berkeley and a PhD in plant pathology from the University of Georgia. After postdoctoral role at Illinois studying plant transformation and defense, he joined USDA-ARS in 2001, where he continues his work on soybean disease resistance. So with that, we're going to start off with you, Dr. Camiletti. Take it away. Thanks.
Thank you. Thank you for the introduction. And hi, everyone. So I'm Dr. Boris Camiletti. I started this position last year. So last year was my first season here in the Midwest. I was in California before. And last year was a good year for diseases. I had the opportunity to see several diseases affecting corn and soybean. Today we will be talking about red crown rot, what we know from the past, and what we are doing at the present, trying to understand, to fill those gaps that we have in research regarding this disease, and how to manage, or some practices that we recommend to manage this disease. So red crown rot is a new disease here in the Midwest, but it's not actually new in the US. It was a very popular disease like 30, 40 years ago in the southern part of the states, in Louisiana, Mississippi. It was affecting peanuts too, so it was popular there in North Carolina, Georgia. And actually, the pathogen had a different name, so the disease was called Cylindrocladium black rot. It was very popular, so there is some research that was done 30, 40 years ago that we use to guide our management, but still we need to validate all those practices here in the Midwest. So this is a disease that is a soil-borne disease. It will overwinter and survive in specialized structures that are called microsclerotia, that are just the spores that are all together forming structures that will remain in the soil and will infect the soybeans the following seasons. So this is a pathogen that will infect the roots right after planting, but has the ability to infect the roots throughout the cycle. Although those infections occurring at the very beginning of the season seems to be the most important ones. There is a lot that we don't know regarding the epidemiology of this disease, although we have some information, as I mentioned, from some research that was done 30, 40 years ago. So it was a very popular disease in peanut and soybean. And so we know that it can be spread within the field. We think that tillage must be the most important factor. Everything that we do to move the soil will move also this microsclerotia, so they will move the inoculum throughout the field. Harvesting, I'm not sure about that in soy, so I included some pictures here, how they harvest peanuts. So you see that the soil movement here is significant, so probably it's having a significant role in the disease when affecting peanuts, but we don't know for soybeans. Planting as well, when we plant, we might move, although in shorter distance, we may move this inoculum also within the field. And from field to field is mainly through the machinery, through the equipment that we use if we plant a field, and then we move this equipment, the planter, without any cleaning to a new field. We might be taking the inoculum with us and contaminating new fields. Also, wind may help spreading this microsclerotia, although it's thought like in shorter distance, affecting the movement of the inoculum in shorter distances. So this is the progression of the disease here in our state. It was first detected in Pine County, and then we were able to keep track of it, analyzing samples, and every time that we receive a new sample, if it was positive, we checked the location and we just highlighted the county. So this is how it progressed throughout the years, or at least how we were able to keep track of it. So understanding how this works, we just receive the sample, we check if it is positive, we just highlight the county, and we keep it highlighted because this is a disease that will remain in the soil. The inoculum will remain in the soil, so the disease will be there for several years. So this, once we highlight the county, we know that the following season you will have a risk of having the disease again, and this is why we keep it highlighted. We don't do like a real-time map because this is a disease that will remain in the soil for several years. So that being said, even when we highlighted all the counties with the same color, the prevalence of the disease is not the same. It seems to be more prevalent in this area based on the number of samples that we receive every year to analyze. This is a picture of the Midwest, how the disease has been reported throughout the Midwest, is now in other states like Kentucky, Indiana, and Missouri. So there are some gaps in the epidemiology that we need to fill with research because we don't know how it moved from here to here if it is only through the machinery, right? So there is also a chance that we are behind trying to keep track of the disease. So if you think that you have the disease, just send the samples to our plant clinic or to my lab, and we can improve our performance in trying to keep track of this disease. These are the symptoms. This is like an early stage for the symptoms on the leaves. So you will see these chlorotic flecks that will occur between veins. And as the disease progresses, these chlorotic flex may continue to develop into interrenal chlorosis and necrosis. So I think Dr. Clough has some images of other diseases that have similar symptoms. This is why sometimes it's difficult to identify. But then if you look at the lower part of the stem and the roots, you will see this reddish discoloration on the stem that is characteristic of this disease. And also, if you see here, we are missing all the roots because they are rotten, so it was very easy to pull the plant. And then only under the right conditions and only during the late stages at the end of the season, you might see these white mycelia with these very characteristic red brick spherical structures that are called perithecia. So this perithecia also has a lot of spores inside, but we don't know actually. We believe that is not based on the research that was done 30, 40 years ago. This seems to be not a significant factor in the distribution of the disease or in the disease cycle. So far, based on the evidence that we have, the disease is mainly spread through the microsclerotia that will remain in the soil and will be moved with equipment, right? So this is the distribution in the field. You will see, like in most of the soil-borne diseases, these patches where you have all dead plants here that are surrounded by healthy ones. So this is very characteristic of the disease. And then if you compare all the yield components, if you take a symptomatic plant and you compare it with a non-symptomatic, and you measure all the yield components, the plant mass, the seed pod count, the seed count, and the seed mass, you will see these reductions. So if we look at the seed mass, we will see reduction of 70%. So those plants will have 70% less yield. So, but actually the impact on the actual yield in a plot will be determined by how big these patches are, right? If you see this scenario where you have a small patch here and the rest of the field looks clean, so you will not see a huge impact on the overall yield. But then probably when you are harvesting, if you go through this part, you will see an impact there. But the final impact on the overall yield is not that significant. On the other side, you may have a scenario like this one. This is a field in Pike County that is severely affected by the disease. They had the disease in previous seasons. And so basically you see like a huge patch. So the impact on the yield here will be very significant, right? So if you walk through this area, you will see plants with no pods, plants with empty pods. So the impact on the yield is significant and will be determined based on how big these patches are. So we are doing some efforts with remote sensing. We are trying to identify the disease using satellites. This is a drone image from a field that we sampled that had like many patches, but the small patches. So we sample and we check if they had a disease or not. So we sample healthy and non-healthy and symptomatic plants. And then we created a model. So the model can help us to identify these hotspots in the field. So we are interested in this because in peanuts and in soybean, like 30, 40 years ago, farmers were able to keep the disease under control using in-furrow fungicide applications. So if we know exactly where the hotspots are, maybe we can target our fungicide applications to these areas, these hotspots, or even we can use a higher dose here. This is why we are interested in knowing where the hotspots are in the field. So if you have a field and you are willing to share the location, please share it with us. We use the location just to take the images from the satellite. We train our model, and then if we validate this model in the future, if you send us the location, we can just take a look very quickly and tell you where exactly these hotspots are. we keep all the information as confidential, so no worries about that. This is a field that had the disease in 2022, and using the satellite images, we were able to identify the disease, and we can see the progress from one season to the other. So in this particular field, we see that the disease increased from 6% to 26% in just one season under the right conditions. So this is a disease that can progress fast, so early detection is important. Here, I just included this to show the timing for the symptoms, because the foliar symptoms will start in the reproductive stages. So normally we don't go and we don't check the stems until we see the foliar symptoms, right? So they can happen more commonly after the R1 stage, but the reddish discoloration at the lower part of the stem will start at the vegetative stages. So this is the first symptom that you can see in season. And then only the perithecia, you will see them very at the end of the season. If you have the right conditions and you may go to the field, you see this perithecia, okay, you are sure that you have red crown rot, but they tend to disappear. So you may go again the following week and all the perithecia is gone. So again, we need to do some research to understand if they are playing an important role in the dispersion of the disease, but it's something that we can use to identify the disease, but also consider that this perithecia may disappear after they are developed. So one of the questions was if we can use foliar fungicides to control the disease, and the answer is no. This is not a foliar disease, so we have the infections only in the roots. What you see on the leaves is like all the toxin produced by the pathogen is being accumulated here and is causing this necrosis. But actually, right now in the market, we don't have any fungicide that can go through the leaves downwards to the roots where the actual infections are occurring. So foliar fungicides, they don't work. And then I mentioned that the infections can occur anytime during the season, but also occurring at the very beginning of the season seems to be the most significant ones in determining the final disease severity. And this is why some seed treatments can suppress the disease. So foliar fungicides don't work, and right now our research is more focused on establishing trials to try all the compounds that we have in the market. We are working together with Dr. Carl Bradley in Kentucky and Darcy Terleco in Indiana, and I think if we combine all our programs, probably we are testing all the products that we have in the market. We are testing seed treatments, in furrow application, 2x2 applications. We are doing some research also in the greenhouse, trying to understand the interaction with soybean cyst nematode, for example. Also nanoparticles, some micronutrients, if they have some, if they play some role in suppressing the disease. Also the inoculum survival, I had some pods here. I think Dr. Clough will talk a little bit about this in his presentation, but we are doing a lot of research supported by the Illinois Soybean Association, trying to find the best management tools for this disease. And that includes also biocontrols. We are starting now our trials. We think that biocontrol may have a chance because if they are able to colonize the roots as the plant is growing, maybe they can offer some protection on top of the protection provided by the seed treatment. And these are some recommendations that we put all together with other colleagues, and it's at the Crop Protection Network. So if you have red crown rot, please make sure that you have the disease. Send the samples to our plant clinic. They can give you a right diagnosis in just a few days. And if you have red crown rot, there are some practices Rotation was recommended at that time, but actually we don't know the real impact on the disease because, as I mentioned, this microsclerosis can survive for several years in the soil. This is a popular disease also in Japan, and they say that this microsclerosis can survive at least 7 years. I had some pots that I think Dr. Clough will talk about these pots later. So again, we don't know the actual impact of the disease, of the rotation on the disease severity. We are not completely sure about that, but it's something that can help to reduce the inoculum in the soil. Also consider using seed treatments. I think right now we have 3 products in the market that were labeled to be used in soybeans against red crown rot. They can suppress the disease, but it's on a state-by-state level. So if you are in other state, you need to check the label to see if it is allowed to be used in your particular state. Also manage other diseases. Soybean cyst nematode. There is evidence from the research in the past suggesting that you may see a higher severity of red crown rot if you have soybean cyst nematodes. We have an entire project that is focused on investigating this interaction, a project that is also supported by the Illinois Soybean Association. So I hope we can have results to share soon. And then it's always a good idea that if you know that you have the disease in one particular field, plant that field at the end during the season. And then after you are done with that, if you want to move the equipment, just wash it on that side to avoid spreading this microsclerosis. So I think I will pass it to Dr. Clough. Happy to take any questions in a few minutes. Thank you.
Okay, this is my first webinar where I'm actually doing something. So patient as I try to figure things out here. Okay. So to share my screen. Right, what do I need to do here?
So on the bottom, there's a little green arrow. You want to hit share?
Okay, share, okay, there we go. Does that work?
Yes, I just put it in presentation mode. We're good to go.
Okay. Okay, so you see it? You have my title slide?
We can see it. It's just not presentation mode, I don't think.
We're seeing the other screen, I think.
Yeah, so sorry. I'm gonna
That worked.
hang
Now,
on. Now as I have multiple screens, I think that's messing me up. Okay, I'm gonna try again here. Okay, how's that?
awesome.
Okay. All right. As I said, first time I actually did this. So thanks for your patience. So this is a presentation I actually just gave a couple of days ago. I modified it a little bit for this audience. I presented at the Soybean Breeders Workshop down in St. Louis. So some of the things are going to be repeated from what Boris said, but I'll go through quickly on those things. So as he says, it's a challenge to identify, especially like drive-by scouting. It tends to be patchy and it looks a lot like other diseases. If I were to ask you what are these diseases here, first one is stem canker. This is SDS, and this is red crown rot. Nice these guys here. Okay. So a lot of things look like red crown rot, just looking at the leaves. So if you want to identify it, you really got to pull some stems and look at the base. And so you have a good sample, you're going to see a nice bright red band right in the crown area. And also, as Boris mentioned, often the roots are fairly well rotted away. In Japan, they actually call this a black root rot. If you happen to see the parathecia, then you can feel pretty confident that you have the right disease. Nothing really makes bright red parathecia like this. As far as the timing, there might be a difference. As on Boris, maybe some of the isolates are producing parathecia a little bit earlier, a little more obvious than others, but you don't always see them. This is a field that I visited in Western Illinois this last summer. And what you'll see is, it's not that obvious, but the ones on the right-hand side, they were full of disease. They had a lot of red crown rot. Whereas the plants on the other side, they looked really clean and healthy. While I was out there scouting, the son of the grower actually stopped by, said, what are you doing in the field? And he was fine with that. I mean, with the USDA looking at diseases and talked about red crown rot a little bit. I asked him about the other side here, why they're, what's different about them. And he said they were planted 3 weeks later, or due to the rain. They had heavy rains out there, so one side got planted, one side didn't. 3 weeks later, they planted this side. And you also note that this side I saw myself was full of flowers, still in the flowering stage, whereas the other side had pod fill stage. And you don't really see the symptoms until the plants start to produce the pods. Okay, the disease is very similar to, or the pathogen, as Boris mentioned, was found on peanut originally and for the US. The first report in the US came in 1966 on peanut. And the disease looks kind of like what you see in soybean. You see the discoloration of the leaves, the wilting, and the roots are all badly damaged and blackened. So they actually call it like a black rot. This article was interesting. This was looking at the first case for this pathogen showing up in North Carolina on the peanut. But they mentioned in here that they also had soybeans growing in the fields nearby, and they were also infected. And that the soybean and peanut isolates, they were pathogenic on both hosts. So the isolates that got off the peanut could have caused disease on the soybean, and the isolate they got on the soybean could infect the peanut. So it seems like there's the same pathogen causes this very similar disease. That's their first report of red crown rot on soybean in the US. I grabbed quickly a disease cycle from Matt Montgomery. His found on the web. I didn't have one myself. And you'll notice that the, starting from here with F, this is where the plant is badly infected. You get the parathecia and microscrotia developed. We're trying to show that microscrotia will come off, they'll persist in the soil. These will infect the next year's crop, and they'll produce these little mycelia that will infect the plants. And then you get the disease. Again, the disease stays in, the disease stays in the roots. The upper part does not have any pathogen. And then it rots, and you go back to the cycle. They show the parathecia down here with the ascospores that they could possibly infect, but there's also a big question mark as to what's their significance in the whole cycle. And then up here was that producing conidia. So does it produce conidia? And the same thing, it's kind of questionable what they really do. Various studies have said that they don't really do much, don't really infect well, but we kind of questioned that. And, I think it should be reinvestigated. So here's a close-up of the perithecia, and you see all these little ascospores being released. And then these cylindrical bundles here, these are the conidia. They're kind of cool looking. These are some kind of vessel that people don't really know what these things do. They might attract insects. But again, the previous publications claim that these are not really that important for the disease cycle. Really important are these other spores called chlamydial spores. They'll aggregate together and they'll form these microsporotia. And the microsporotia seem to be the main source of inoculation, of inoculum. So the fungus was also, the disease was found in Japan back in 1969. They had on soybean. And since 69, the Japanese have actually been doing a lot of research. So a lot of the papers actually come from Japan on this pathogen. One of the contributions they made that was kind of significant was showing that the microsporotia could survive in the soil for a long time. So they show that, up to at least 7 years, they're still detecting infectivity on soils that had microsporotia. And then one could say, well, that's Japan. What about Illinois? So Boris mentioned his pots. So these pots came from Nathan Klashevsky. When he was here, he set up these pots with inoculum, and then he left. He went to industry, and the pots just sat there until Boris showed up. And so Boris planted soybean in these pots, and in some of the pots, he got the red crown rot disease again. So that was showing that the pathogen can survive the Illinois winters for at least four seasons. Okay, how do they spread? And again, Morris is saying that it seems to be short distance spread, mainly through soil movement. The spores, we're not sure they really have a big impact, but there could be some spore movement too. But the main thought is that it's by microsporotia going short distance through soil movement. Could be some debris from the harvesting. The farm equipment seems to be major. When I went scouting with Robert Bellum, who's down in Madison County, he was telling me that, often you just look to where the farm equipment enters the field, and that's where you'll find your red crown rot initially in the field. Maybe not always, but often. And then also people and wildlife going through. Robert was thinking one of the factors could also be waterfowl. If they come in through from one field to another, they might transfer some of the microsporotia. And then water movement, if you happen to be in an area that has some water runoff. Okay, as Boris showed, he had some satellite imaging showing how the disease does spread. It does spread around. So from one soybean season 2022 to the one in 2024, he had an increase from 6 to 26%. Oshii and all in Japanese group estimated maybe about a 30% yield loss, whereas the group from Carl Bradley recently said maybe it could be up to 60%. This is a field, I got a slide from Carl Bradley the other day. He presented this at the soybean breeders workshop. That should be SBW. So what they did, he had permission to, on a research plot in Kentucky, to plant strips of where one strip, two rows were inoculated and two were not. And so then he harvested the plants from the rows that were inoculated and did some yield estimates. And then he did the same thing with the rows that were not treated and did a comparison. And when you did that comparison, what he has on the x-axis is how much damage do you see on the leaves, a foliar disease index. The more you have disease index showing up, the greater the yield on the y-axis. So he was showing that he was getting close to 50 to 60% damage or yield loss when you had the most badly damaged plants. A group in Japan also looked at the infection process in the roots. So they put the roots in these little bags and having fungus infect, and did a histological study, looked with the microscope. And it's not super obvious, but the cells here in this area here are going through the epidermis of the root, and you can see a lot of mycelia in there, and then they don't get through here. this is where they say the paraderm is starting to form. So as roots get some suberin development, they're more resistant. And what they concluded with this too is that, and this is supported by other studies, that the most vulnerable points are the young seedling before the roots have that nice strong paraderm on them, and then growing root tips, wounds, like Boris said with nematodes, and adventitious root emergence. I don't have the slide here, but I was looking up some stuff just before talking. The group in Louisiana was studying this, and they were showing that if they inoculated the plants within the first week, they got the most severe symptoms later on. And if they inoculated like 2 weeks, 3 weeks, 4 weeks later, they still got some infection, but it was like half what they got if you get inoculated in the very, very first week. So that very first week seems to be the most critical point, which also kind of fits what Boris was saying. If you have some biocontrol at that very first week, that might help, as well as seed treatments. Don't let the plants get infected during that first week or two. As mentioned, Louisiana, they had red crown rot detected there in 1976. And they've done a lot of work on this disease. Here's an example looking at the effect of soil temperature. So they were showing that once the temperature got up really pretty high, say 35 or more, they didn't see a very good infection at all. And they also showed that if Louisiana, of course, super hot. So if they were to delay their planting into middle of June or so, when the soil temperatures got really hot for them, they saw much less disease. It went down drastically. Carl Bradley was mentioning that the optimal temperatures for infection, if you go in Fahrenheit, be about 77 to 86. And so maybe for our area in Illinois, it could be that the earlier planting when the temperature is too cold, maybe you don't see as much active infection if it's too cold for the fungus. There are optimal range of soil temperature for the fungus to be active and for infection to occur. Okay, so the pathogen Polynectria leucicola has been found all over the place. It infects many different plants, infects peanut, soybean, blueberries, papaya, eucalyptus, many things, blueberries. But only recently showed up in Illinois. A paper by Nathan Klaschewski. This just came out in 2017 is when he actually did the isolation. If you look at what Carl was saying, where was the disease in 2014? So Nathan found this in 2017 in Illinois, but prior to that, there wasn't much red crown rot around. And there was some in Louisiana, South Carolina, North Carolina, Virginia, maybe some in Georgia too, but down in the deep South. Okay, so when I started this project, it was because many of you may know Glenn Hartman. He was the kind of the main soybean disease expert in the US. And he retired a few years ago. And he's in my research group here with the USDA on the University of Illinois campus. And I was asking Glenn, what were some things that maybe I should look at? And he suggested red crown rot. At the time, he said, it's only been detected in a couple of places, but, it might disappear or it might, spread. So he recommended that we try to characterize the isolates and try to see if it's a lot of variation, like it's been there forever, or little variation that would suggest that it was introduced. And then how does it compare to some other isolates that might be in the repositories? So we collected in 2022, 2023, and 2024, from 27 to 39 isolates each year. And we also got some from the repositories, and these were deposited between 1970 and 1988. We got from Georgia, North Carolina, South Carolina, Louisiana, off of peanut, blueberry, and soybean. So what we did is people have recommended certain targets, genetic targets. You can do PCR and get the sequences of those regions and then compare them to see how much variation there is between your isolates and see how similar they are. So as Boris was showing a map of where it was been reported as of 2024. And these are where we got our isolates. So some of these we did ourselves with scouting, and some we got from some collaborators. And what we found is instead of a lot of variation, we saw very little variation. But we see there might be two closely related isolates in Illinois. And for actually for this whole region, we only found two different isolates based on these 6 PCR targets. If you do more sequencing, we might see more differences. So these Illinois isolates are different from these historic because they peanut isolates. The historic isolates, though, are actually all very similar. They all have the same DNA sequences for these 6 targets. So don't have the same pathogen that was out, spreading around that time. But it looks like it could have been. Yeah, this table summarizes the genetic findings. So basically, we have these different PCR target regions, and we sequence them, and we see which base might have been different. And so if you look at the ITS region, this set had 1 T, this set had 2 Ts, this set had T and actually more stuff. So for each of these, we had some kind of they call it polymorphism, some kind of a difference. The isolates in Illinois, depending on where they came from, they only had the difference in the ITS region for these markers, whereas compared to the historical isolates, they had a lot of differences. So you see the ones that came from Pike County and from Kentucky were actually very similar, and the rest of Illinois was another group. I didn't put the ones from 2024 in here because it basically follows the same pattern. And I'll summarize that in a figure. Before that, I just want to show, in case you're curious about how people can see the DNA differences, the sequencer gives you these graphs, these chromatograms, and you can just count the peaks here. This set here has 7 very clear peaks, whereas this set had 8. The sequence beyond here is all the same. The sequence beyond here is all the same. So you can identify differences. And then this is the historical isolates, ones from the peanuts. You see they're very different in this region. Again, back here is all the same for hundreds of bases. Back here, hundreds of bases all the same. But this region is all kind of different. Okay, so a summary of that would be, sorry, the ones that had the 8 Ts came from Pike County and some neighboring counties, as well as ones in Missouri, Indiana, and Kentucky, whereas the rest all had the 7 T isolate. And down here in St. Clair County, we saw both of them. Now, some of this is due to how much collection we do. Some counties, we have many, many isolates, more than 10, you say, different fields. And some, like in St. Clair County, we only had 2. So 2 different fields. One field had the 8 Ts, one field had the 7 Ts. Okay, so the main points, for my presentation would be that since 2017, red crown rot has been verified in Illinois, Indiana, Kentucky, and Missouri. The symptoms are most likely only after the flowering period. Spread is mainly by movement of the soil or plant debris. The main infection structure are aggregates of chlamydospores that form these microsporotia. Microsporotia can persist in the soil for up to 7 years, at least. Preliminary data of the isolates collected in the Midwest suggests only 2 genetically different isolates. And the preliminary data of the isolates collected in the Midwest is consistent that the pathogen was introduced somewhat recently. And then thanks to people that helped. Teresa was actually a technician with Glenn Hartman. She has a lot of experience working with fungi. She isolated all the, purified all the isolates. Nancy McCopen has also been with the USDA here for a long time. Just worked with Les Domeyer. She did all the DNA work and all the PCR sequencing. Student Samuel and postdoc Juan, they did all the phylogenetic analysis. And then thank to Robert Baum, who provided our first samples. He's the one who showed me the first fields down in Madison County. And Robert knows a lot about this disease. And then Stephanie, of course, she's helping to organize growers and send samples to the University of Illinois and to the USDA here. Ready for questions?
Do you want to go ahead and take the first one, or we can maybe expand on the first question from Kyle? What fungicides are the most effective seed treatments or in furrow? Can you make any comments on that, or do you want to make any comments on any products that are currently labeled for red crown rot?
Yeah, so the Inforo is under research. We are running the trials. We started last year with the trials, so we need several years to get good data. The industry is also doing some supporting those trials. So Inforo application might be an option in the near future if we find some products that may help. Basically, because we are spreading or covering much more area than just with a seed treatment, right? So we think that we can protect the plant from other infections from inoculum that is kind of far from the seed. And then with the seed treatments, we don't have data on many of the products. We know that Saltro is suppressing the disease based on some data that Dr. Bradley shared with us. Is suppressing the disease, is not a complete full control, and it will depend also on the location and the year, because the weather conditions may affect also the performance.
And another question came in previously, so I just wanted to make sure that I did, because I've been hearing about it myself, that Olivo now has a 2-EE suppression as well for red crown rot. But I did just look up the label, and I do want to make a comment that that labeled rate would be different than what maybe you might be used to using for Olivo for Sundance syndrome. And so it's 0.25 milligrams of AI per seed is the recommended labeled suppression for red crown rot with Olivo, where 0.15 milligrams of AI seed would be what is labeled for SDS. So, do you have any comments about the new Timerium? It's new from Syngenta. I don't think it's even been released yet. Are you working with, are you getting to work with that and with your research?
We haven't tested that product in the past, so but I'm hoping that we can test it this year. It's not commercial, I understand.
Okay, then here's the other big question, and this is a good one, Jim. Are there any known broadleaf weeds that could serve as hosts? Do you guys want to make any comments on hosts?
We know alfalfa. I'm thinking of crops here, right? I mean, there are a lot of ornamental plants and blueberries that can be a host, but they're not that common here. I don't know if the weeds is something that we need to explore. I will be working with Dr. Hager. He will give me like a bank of seeds so I can see if they get infected or not, if there may be a role there.
Yeah.
Yeah, the Japanese published something, I forget what they are. Grasses tend not to be host, but for broad root leaf weeds, I'm not sure about that one, which ones might be in the fields here that might be host.
But it does have a fairly broad host range, and it's not just legumes. So
Right. You got eucalyptus trees, papaya trees, blueberries, peanut.
I'm gonna go back and see if I can get your comments to previously. It was mentioned about, and I know you went over how we want to try to, I say we because I really want to know as well, but look at what are your thoughts on, do you think there's a potential resistance difference between different soybean varieties? And again, I know Boris is working on this, but just comments on that.
In the publications, like down in LSU in Louisiana, they have a couple papers showing some varieties working better for them. When we tried doing the testing ourselves, we're just not getting good, setup or something. We're having a hard time getting consistent results. So, and then in Japan, they claim to have some that were better than others. But when I talked to the researchers, they said it was kind of not real strong. But I would like to try to do some in field studies, just get a better feel for if something really is resistant or not, because we're just having a hard time getting good results with the lab-based assays.
And I don't, Dr. Camiletti, I'll let you comment too. And I was just going to add, I think there was also a question about different maturities, but I don't know if we have enough information on that as well.
I don't have enough information about the maturity groups. What we will do this season is we will try some genetics that we have at the university, trying to see if we have better or worse performance in some of them. I think I will have other commercial varieties too from some companies, but I know that other companies are working on trying to test these varieties for red crown rot. Just anecdotal information, I've seen some differences in our trials, so we need to explore that as well.
Okay, go ahead.
I heard quite a bit from the company that they're actively, screening their own germplasm, and some of them claim that they have some good stuff.
So one thing that's talked about a lot is different planting dates. We tend to plant, or I should not speak for everybody, but a lot of people plant earlier into cooler soils. Do you have any comments on that with this disease, like for different planting dates and also in relation to disease pressure?
And so the one study from LSU is showing that the worst time to get infection is a very early stage. So if you get, fortunate with the weather conditions and you plant early, maybe it's going to be too cold up here to get infection in the very first week or two, and then you might get much less disease. You still get disease, but it'd be much less.
Yeah, I think we need to think on that, like considering the temperature, the soil temperature, more than the planting date. If we know that the pathogen is happy and able to infect the roots, optimum temperatures are 77 to 86 Fahrenheit. Probably we want to avoid those temperatures during planting. Here in the Midwest, Dr. Bradley is doing some research on this, but it seems to be the opposite trend, like compared with the southern. So later planting seems to be more conducive for this disease.
I was going to say, I don't have very many observations, but I hate to be the Debbie Downer in the room, but I've seen fields, of course they were different varieties, but side by side, one planted early, it had red crown rot, and one planted later, and it had red crown rot as well. So I don't want people to think that just because you're planting earlier, you can avoid the disease. I do think the symptoms progress more quickly. Maybe, yeah, go ahead.
So that the study in LSU was saying that they still got disease, and so they had like maybe half as much the severity. The severity was like twice as much in the ones that got infected really early in that first week.
Yep. Okay, here we go. What else? There's lots of questions here. Oh, crop rotation. What are comments on crop rotation? Is that going to help hinder the disease?
Well, maybe I think that we got that recommendation based on research done in the southern part of the state, where probably they have more crops to rotate than we do here. So if we consider that the inoculum will remain in the soil viable for several years, we don't know the actual impact of the rotation. I know that farmers are going to continue corn because of this, so we may get some information from those farms. But I don't know, Steve, what you can comment on this.
I haven't seen things specific for soybean. So the studies are out there. I haven't seen them. But in peanut, they were saying that a 2-year rotation, like skipping 2 years with a non-legume, non-host plant, was the same as skipping 1 year. So they saw they got a reduction if you should never plant, continual soybean. And so with peanut, if they plant continual peanut, they got a lot more disease. And if they had 1 year of crop rotation with a non-host, they got a lot less disease. And then they said a 2-year made no difference from a 1 year. But then our colleagues in Japan were saying that they do a rice rotation, which is very different because they flood the field for the rice. But they found that a 2-year rotation worked better than a 1 year. So that's another study that still needs to be done, I think, unless we just missed it in the literature.
One thing that's come up, and I know we've all talked to some of the farmers that they probably are, dealt with this disease, especially in areas like Pike and Madison. And one of the things that's come up is that they wanted to know about high pH or soybean cyst nematode with the disease. And I know, Boris, you're doing some research on the SEN. What about pH? Do you think that could affect this disease?
That's another factor that we need to investigate. Yeah, I mean, the pH is a factor that can influence infections, fungal infections. So may play a role, but we don't know if there is an optimum pH for this pathogen with the soils that we have in Illinois. It's something that definitely needs to be investigated.
I'm looking through all the questions that were sent in, and they were great, by the way. And I know most of you have covered most of this. And we hit on Olivo. That was a common question. I guess there was questions about, do you think there's a high amount of seed treatments that's been used in Illinois? We're known, and I'm not saying other states don't use seed treatments, but we really like our seed treatments here. It seems like there's a high use of either the two main ones, and I don't want to say that we're not using other seed treatments, but we're using Saltro and Olivo for SDS and maybe some SEN suppression. Do you think that those possibly could have been masking red crown rot? That's one question. Or do you think we still get symptomology when we use those products?
So it's hard to say.
Yeah, I think these are all like hypotheses. One of them is like, what if these products are really good killing nematodes and sudden death syndrome, and probably this pathogen found like a free way to infect, because it seems to be like a bad competitor actually, right, Steve? Based on what we see in the lab when we try to isolate the pathogen, and we have other organisms there, will outcompete this Calonecria. So
it grows slowly.
but it's just an hypothesis. We haven't done research.
Yeah, but it might have been here for a while, because this tends to be patchy and it looks like SDS or something else, and people might ignore it. So, it spreads into a bigger area of the field. But it probably hasn't been here for a super long time, I would guess.
Yeah, and kind of cool with other diseases too. You may have red crown rot and sudden death in the same field. We had near Bloomington, we had a field with white mold and red crown rot together at the same time. Definitely not a good combination to have. Can occur also with other diseases.
Oh, that's a good point. I know in my experience when we went out to the field, especially late in the season, that it often shows up with other diseases, like you said, and that's good. And it's also hard to tell because a lot of the plants could have stem canker and SDS and red crown rot at the same time.
Right.
Yeah.
So really good point, Boris. One last question here. Is any testing being done with red crown rot and Peking traits to help control SEM pressure?
Yeah, not from our group. The Boris, you have any that are like known to be SEN resistant in your trials?
Any what? Sorry, I don't think I understood the question.
Like Peking has SEN resistance, so using an SEN resistant variety might give you some better resistance to red crown rot due to less nematode damage.
No, but we will definitely explore that in this project that we have with supported by the Illinois Soybean Association. Yeah.
Okay.
There is so much research that needs to be validated here in the Midwest.
So many questions, so many unknowns. So I know, again, thank you so much to both of you. I really appreciate both of you and like working with you. So I can't wait. I know Boris and Steve, you're both doing a lot of research, and we'll keep continuing to have updates so we can get answers to some of those questions. So thank you so much.